Good day, everyone. I'm Sir Franz, and welcome to our lesson on the Krebs cycle. The Krebs cycle is also known as the tricarboxylic acid cycle and the citric acid cycle. As you will observe later, as we show the structure of citric acid, one will understand that it is a tricarboxylic acid, and they refer to the same thing. The Krebs cycle is the final common pathway for the oxidation of carbohydrates, proteins, and lipids or fats, which eventually forms acetyl coenzyme A, which is then catered by the Krebs cycle and oxidized. The Krebs cycle occurs in the mitochondrial matrix, which is the liquid portion of the mitochondrion, the innermost portion of the organelle. At the end of the day, the Krebs cycle will produce carbon dioxide and reducing equivalents. Now let's turn our attention in the image in the right. One will see that carbohydrates, through glycolysis and the intermediate step of pyruvate dehydrogenation, produces acetyl coenzyme A, which has two carbons. Proteins, through deamination, will also produce acetyl coenzyme A, and lipids, through beta oxidation, will produce acetyl coenzyme A. In general, acetyl coenzyme A fuses with the 4-carbon molecule oxaloacetate, and together with water, it will form citrate, which is the reason why the cycle is called the citric acid cycle. Through the cycle, the carbons that are contained in citrate are oxidized, combined with oxygen, to liberate carbon dioxide. But at the same time, these processes produce reducing equivalents. Reducing equivalents are in the form of reduced NAD and reduced FAD, the two molecules that carry hydrogen into the electron transport chain, or oxidative phosphorylation, which will eventually form a chemiosmotic gradient that powers the production of adenosine triphosphate. So the end goals of this pathway are to produce carbon dioxide and at the same time, more importantly, produce the reducing equivalents, which are the major energy sources. Let's now go into the reaction steps. The image in the right can be quite overwhelming. And so let's just focus our attention first on the text in the left. So in summary, the reactions of the Krebs cycle are the following. You have to first combine acetyl coenzyme A with oxaloacetate to form citrate. Citrate is then isomerized, which means the number of carbons and the number of oxygen and the number of hydrogens, in general, the number of atoms remain the same. It is just the connection between the atoms that are made different. That's what isomerization means. So citrate is isomerized and then oxidized. And the product of that will be ketoglutarate or alpha ketoglutarate. Alpha ketoglutarate is then oxidized and then that forms succinyl coenzyme A. Thiolysis of succinyl coenzyme A occurs, which means that the CoA molecule is separated from the succinate molecule, and therefore the product will be succinate. This releases a lot of energy, and therefore, at this step, substrate level phosphorylation occurs. Step number 5, succinate is oxidized to form fumarate. Fumarate is dehydrated, which means you remove water to form malate. Lastly, malate will be oxidized and that will yield oxaloacetate, which is what participates in the first step of the cycle as it combines with acetyl coenzyme. And therefore, what does this tell us? We start with oxaloacetate and end up with oxaloacetate. And therefore, That is a cycle. So let's now turn our attention to the image in the right. Again, that is the structure of acetyl coenzyme A, which we have discussed in the intermediate step of glycolysis. The bond between acetyl and coenzyme A is a thioester bond. That is why we will see the sulfur atom there. So, acetyl coenzyme A combines with oxaloacetate and that will form citrate. But at the same time, the coenzyme A is liberated. So that is the structure of citrate. You have here a central 3-carbon molecule, and each carbon binds a molecule of a carboxyl functional group. So you have 3 carboxyl groups, COO, COO, and COO. That's why it's called a tricarboxylic acid molecule. Next, citrate will then be isomerized first into cis-aconitate, which basically occurs when water is removed from citrate. You then add the water back through the enzyme aconitase, which is a form of hydratase, which means it will add water. And then you will be able to form isocitrate. So why is it called isocitrate? Because in citrate, the hydroxyl group is found in the central carbon, while in isocitrate, the hydroxyl group is now found in the terminal carbon. Next, isocitrate will undergo the first dehydrogenation reaction. And as what we have already discussed, dehydrogenation involves the removal of hydrogen. That's an oxidation reaction. And therefore, it should be coupled with a reduction reaction. And the one that is reduced is NAD. And as NAD is reduced, it will take up hydrogen, and therefore it will produce NADH, or reduced NAD. But also at this step, as you will observe, it will form a highly unstable group. And therefore, that highly unstable group, the carboxyl group in the middle, will have to be cleaved off as carbon dioxide to form alpha-ketoglutarate. And so, the oxidation of isocitrate to alpha-ketoglutarate actually involves 2 steps. First, simple dehydrogenation, and then the cleavage of the carboxyl group to liberate carbon dioxide. And so this step will produce NADH and carbon dioxide. Alpha-ketoglutarate then is oxidized by the alpha-ketoglutarate dehydrogenase complex, which is similar in structure and active groups as pyruvate dehydrogenase, which is the intermediate step of glycolysis. So alpha-ketoglutarate will then undergo a dehydrogenation reaction, and therefore you will be able to produce at this step also a molecule of NADH and carbon dioxide. This step is specifically inhibited by the poison arsenite, which is a derivative of arsenic. So next, because this dehydrogenation reaction creates a highly energetic functional group, which is also unstable, this will have to be stabilized by the attachment of a coenzyme A molecule. And therefore, the end product of alpha-ketoglutarate dehydrogenation would be succinyl coenzyme A, which already contains coenzyme A, or SCOA, in the structure here. But at the same time, like isocitrate, we will be able to produce another reducing equivalent in the form of NADH and carbon dioxide. So at this step, the 2 carbons that were added into oxaloacetate have already been removed as carbon dioxide. Next, we have to liberate the high energy bond between coenzyme A, and that is through succinate thiokinase, which will remove the coenzyme A, but at the same time capture the energy that is in the coenzyme A and transfer it into ADP to form ATP. That is a form of substrate level phosphorylation. But in other texts, you will see that the one that captures the phosphate group would be GDP. And therefore, GDP forms a GTP, guanosine triphosphate, but at the same time, this is equivalent to a molecule of ATP. So 1 molecule of guanosine triphosphate is energetically equivalent to 1 molecule of adenosine triphosphate. That step will produce succinate. Then the next step will be to again oxidize succinate. That is through a dehydrogenation reaction, but this time, the participating reducing equivalent is not NAD, but FAD. So at this step, you will produce reduced FAD or FADH2. This step is very important and it's very unique because this step, even if it is a part of the Krebs cycle, is at the same time a part of the electron transport chain, which we will discuss later on. The end product of this stage will be fumarate with that structure. There's a central double bond between the two carbons, and each central carbon will hold 1 molecule of carboxyl group or COO. And then fumarate will be hydrated at the central double bond to form malate. And lastly, malate will then undergo the third dehydrogenation reaction, and that is through the third dehydrogenation reaction that uses NAD, and that will form NADH. Then that will reform oxaloacetate, which is the starting point of this cycle. So again, let's recapitulate. The products of the reaction will be 10 molecules of ATP per turn of the cycle. This value of 10 is derived from the production of 3 NADH molecules, which if oxidized in the electron transport chain, will each liberate 2.5 ATPs, 1 molecule of FADH2, which at the electron transport chain will produce 1.5 molecules of ATP, and 1 molecule of GTP or ATP. And that will have a total of 10 molecules of ATP. It is important for us to take particular note where in the cycle carbon dioxide leaves. So, carbon dioxide is liberated from the oxidation of isocitrate and from the oxidation of alpha-ketoglutarate. Where in the cycle are the 3 NADHs liberated? 1 NADH will be liberated from the oxidation of isocitrate. Another NADH will be liberated from the oxidation of alpha-ketoglutarate. And the last NADH will be liberated from the oxidation of malate. The FADH2 or the reduced FAD is liberated from the oxidation of succinate into fumarate. And the 1 GTP or ATP is produced by a substrate level phosphorylation from the thiolysis of succinyl-CoA into succinate. As what we have emphasized a while ago, there are 2 carbon dioxide molecules that leave the cycle, while there are 2 carbon atoms from 1 molecule of acetyl coenzyme A that enters the cycle. And so, we have a conservation of the number of carbons in the cycle. The Krebs cycle is termed amphibolic because it participates in both catabolic and anabolic reactions. Again, catabolic reactions will degrade a larger molecule and produce smaller products, while anabolic reactions will fuse together smaller reactants to make a larger molecule that is the product. So, the Krebs cycle has a central role in gluconeogenesis, which is the formation of glucose from non-glucose sources. It participates in lipogenesis, which is the synthesis of fat, and in the interconversion of amino acids. These key points are illustrated in the image in the right. Again, pyruvate is the end product of glycolysis, and through pyruvate carboxylase, we will be able to form oxaloacetate. And this step is what happens in gluconeogenesis. Oxaloacetate leaves the cycle as phosphoenol pyruvate through the action of phosphoenol pyruvate carboxykinase. And PEP will then go up the steps to form glucose through gluconeogenesis. Lactate can liberate or produce pyruvate by the removal of a hydrogen atom, and pyruvate can be produced from the deamination or the removal of the amine group from these amino acids: hydroxyproline, serine, cysteine, threonine, and glycine. For you to better appreciate these amino acids, kindly search for their structure and why pyruvate can be liberated from their structures. Tryptophan associates with alanine through alanine aminotransferase, and alanine, again, can produce pyruvate by the removal of its amine group that is catalyzed by transaminases. Oxaloacetate can communicate with aspartate, which is another amino acid, through a transamination reaction. So what does that mean? If you remove an amine group from aspartate, we'll be able to form oxaloacetate, which can then enter the Krebs cycle. Glutamate can communicate with the Krebs cycle by transamination, which means if we remove an amino group from glutamate, we will be able to form alpha-ketoglutarate, which is a participant of the Krebs cycle. Propionate, which has 3 carbons, can be mutated or transformed in structure and then enter the Krebs cycle through succinyl coenzyme A. This step right here, propionate to succinyl coenzyme A, requires vitamin B12 or cyanocobalamin. Isoleucine, methionine, and valine can enter the Krebs cycle via deamination and enter the cycle as a molecule of succinyl coenzyme A. Tyrosine and phenylalanine, which are amino acids with a benzyl ring, can enter the Krebs cycle as fumarate. So, what does this image tell us? That the different amino acids in the body can produce molecules that eventually enter the Krebs cycle. And because we can take oxaloacetate away from the cycle and produce phosphoenol pyruvate from it, technically, these amino acids which can enter the Krebs cycle can also produce glucose. And therefore, we have a way of producing glucose from amino acids. And that is the process of gluconeogenesis. And therefore, the amino acids that you see in this diagram are termed gluconeogenic amino acids. Again, because they can form molecules that are part of the Krebs cycle or enter the Krebs cycle, and because oxaloacetate can be taken away from the Krebs cycle to form glucose. Next, as you have observed, there are a lot of reducing equivalents formed from the Krebs cycle. And these reducing equivalents, if oxidized in the electron transport chain, can liberate a lot of ATP. And therefore, the Krebs cycle has a central role in energy production. Therefore, it is important for the body and the mitochondrion to be able to regulate the Krebs cycle in response to decreasing levels of cellular energy. So how does the body regulate the Krebs cycle? The first point will be the levels of energy-carrying substances. So low levels of NAD, which means that there are low levels of energy, and high levels of ADP, which means that we have low levels of energy, will both speed up the Krebs cycle. Dehydrogenases, which are enzymes in the Krebs cycle, are activated by high levels of calcium in the cell. So what does that mean? For example, in neurons, which will eventually release the neurotransmitters in response to high levels of calcium, would also activate the Krebs cycle so that there is enough energy for the function of the neuron in neurotransmission. Again, calcium, high levels of calcium in the cell will activate dehydrogenases of the Krebs cycle. Another example will be in contracting muscle. Remember that calcium causes muscular contraction. Therefore, when the muscle contracts and utilizes a lot of cellular energy, that calcium, aside from starting muscle contraction, will also actually speed up the metabolic pathways that produce ATP, so that you have sufficient energy to power the contraction or sufficient energy to support neurotransmission. Dehydrogenases, again, which are enzymes in the Krebs cycle, are also activated by low levels of ATP. Citrate synthase, which catalyzes the combination of acetyl coenzyme A with oxaloacetate, are inhibited by ATP, which just tells us that if we have high levels of ATP or cellular energy, Krebs cycle will automatically slow down. Long-chain fatty acids, which are products of anabolic reactions or the building up of larger molecules from smaller molecules, this time acetyl coenzyme A, combining with each other to form long-chain fatty acids, will also inhibit citrate synthase. And what would that tell us? That if we are already making fats because we have excess energy, we have to slow down the Krebs cycle because we have sufficient energy. And therefore, long-chain fatty acids will also slow down the Krebs cycle. So in the image here in the right, we will understand that NAD, AMP, and coenzyme A, which are indicators of low energy, will speed up the production of acetyl coenzyme A from pyruvate through the intermediate step. Next, ATP and citrate will slow down the Krebs cycle. NADH, or high levels of the reducing equivalent NADH, will slow down the Krebs cycle. ATP will slow down the Krebs cycle. ADP, which is an indicator of low cellular energy, and NAD, not the reduced one, also an indicator of low energy, will speed up the cycle. So you will understand here that if we have conditions inside the mitochondrion which relate to high energy levels, it will slow down the Krebs cycle. And if we have conditions in the mitochondrion that relate to low energy levels, such as ADP and NAD, it will speed up the cycle. So that is a form of negative feedback. And lastly, let's discuss the clinical application of the Krebs cycle in hyperammonemia. So, remember that ammonia is a toxic product. Particularly, ammonia is produced from the deamination of amino acids to liberate energy. So, the body would have to find a way to control the levels of ammonia, again because it is toxic. So the body converts ammonia into urea, which is eventually urinated and will be excreted by the kidneys. So in that way, the body removes excess ammonia, converts it into urea, which is then eliminated through the urine. So in a normal setting, urea is eliminated and ammonia does not accumulate. But accumulation of ammonia can occur in abnormal states if you have disease, such as reduced functional liver cell mass. So, for example, if you have hepatitis, where there is inflammation of the liver, those liver cells will not be able to catalyze the formation of urea. If you have a disease that leads to chronic destruction of the liver and therefore low activity of the liver cells, for example, in cirrhosis, where there is substitution of the liver cells by fibrotic tissue or scar tissue, that will lead to the accumulation of ammonia. Also, there are genetic defects or inherited defects in the urea cycle. The urea cycle is how urea is formed from ammonia, which we will discuss in later lessons. There are certain individuals with genetic lesions that preclude them from performing the urea cycle, and therefore they cannot form urea, and therefore in these individuals ammonia will accumulate. So, if the individual will not be able to form urea to lower down the levels of ammonia, the body will resort to an alternative way of handling ammonia. And that is through the conjugation of ammonia with intermediates or participants of the Krebs cycle. So, what does that mean? High levels of ammonia will then be taken up away from the blood into the mitochondrion and added into the participants or the intermediates of the Krebs cycle, more importantly, alpha-ketoglutarate. So, if we will turn our attention in the image here in the right, we will see the interaction between the neuron, the glial cell, which is the astrocyte, and the capillary cell or endothelial cell. So ammonia in the blood will pass through the endothelial cell and stay in the astrocyte. That ammonia, NH3, will then take up a molecule of alpha-ketoglutarate, which again, if you will remember, is an intermediate in the Krebs cycle. Ammonia combines with alpha-ketoglutarate to form glutamate, which is an amino acid, and glutamate again conjugates another molecule of ammonia through glutamine synthase and forms the amino acid glutamine. So, through that process, you have decreased the levels of ammonia in the blood, but at the same time, decreased the levels of alpha-ketoglutarate in the Krebs cycle. Eventually, the low levels of alpha-ketoglutarate will stop or slow down the Krebs cycle and therefore slow down the production of the reducing equivalents, leading to low levels of cellular ATP. Because the neuron is a highly energetic cell and the central nervous system is a highly energy requiring organ system, high levels of ammonia, which again decreases the levels of alpha ketoglutarate and truncates or stops the Krebs cycle, will then manifest as neurologic signs and symptoms. That means the nervous system is affected. And how does that manifest? The patient will have lower levels of sensorium or altered sensorium. That patient can be irritable. That patient can be confused. The patient can be in a state of coma. There will also be altered reflexes such as asterixis. If you will Google or look at a YouTube video of asterixis, that will show you a video of tremors in the hand, a particular set of tremors that involve the wrist. That is what we call the liver flap. So that is a form of altered reflex because of abnormal regulation of neurons that control the wrist and muscles that activate the wrist. Okay, so at the end of the day, ammonia is toxic. The liver handles it through formation of urea. If you can't form urea, ammonia will accumulate and take up alpha ketoglutarate from the Krebs cycle, which will decrease energy in the neurons and therefore cause neuronal dysfunction. Okay, so through this lesson, we have discussed the Krebs cycle, the actual reactions of the Krebs cycle, what goes into the Krebs cycle in the form of acetyl coenzyme A and the different amino acids, how the Krebs cycle participates in gluconeogenesis, lipogenesis, and the interconversion of amino acids, how low levels of cellular energy will activate the Krebs cycle, and how high levels of cellular energy will slow down the Krebs cycle, and how ammonia is devastating, especially to the nervous system. So I hope you learned a lot through this lesson. Thank you for listening.